So once protein synthesis is done and we're done doing transcription and then translation, there are usually a couple of things that need to happen to get the protein in the correct shape and then get it to the correct place that it needs to go. This is called protein processing. There are chaperones. If you remember going on field trips, you have chaperones that come with you. Chaperones are proteins inside of the cell. They're going to help bring the ribosome mRNA complex during translation over here to the rough ER, right? So this is the rough ER, which is made for secreting proteins. The rough ER, we're going to end up with that protein kind of inside of the rough ER here. And what's going to happen is once the chaperones have let it here, we're going to do something called post-translational modification. So this is a really long word, and let's break it down so it's not so scary. So what does the word post mean? Well, post just means after. So it is after what? After translation. So that first word, post-translational, just means we just did translation. That's all that it means. And then modification just means changes. So all that's happening really are changes after translation. So sometimes those proteins come in as a long string and they need to be folded up properly. And a lot of the times we'll need to add some carbohydrates on here, other macromolecules to the protein so that it is the right shape so that it can function properly. So I want you to think about this. Do you need to copy all of your genes into proteins all of the time in your body? Hopefully you're shaking your head no. It would be kind of ludicrous to have all of our genes turned on making proteins at the same time. So we don't always need that process of protein synthesis to be going on. So gene regulation is the idea that, so genes are kind of like a light switch. They can be turned on or off depending on if I need the protein that they make. So here is a gene protein pathway for the protein prolactin. And this is the hormone that stimulates mammary glands to produce casein. And casein is the protein in breast milk. So does a person need to be producing casein all of the time? Absolutely not. Only a mom who's actively breastfeeding her child needs to be producing this protein, right? So what will happen is the hormone tells the cell, hey, we probably need more breast milk. And the thing that stimulates this hormone is actually baby feeding. So this is why the more you feed the baby, the more milk you make. Because when the when the infant suckles, you produce this hormone prolactin. So it empties your milk and it tells you, hey, we need to make more milk. So we goes down to our DNA and says, we need to make, here's my gene for casein. I need to make the RNA. My RNA needs to make some proteins, and then those proteins are going to get secreted out as casein, and then they can go to the blood supply, get filtered out into breast milk. When a baby stops breastfeeding, or mom decides to stop, this prolactin hormone stops getting stimulated, and so we stop producing casein, and then you stop producing breast milk. So take a moment and reflect on the post-translational modifications of proteins.